Hey, welcome back to the channel. Today we want to talk about some things from developmental biology. I don't think I've talked at all about that on this channel. So we want to talk a little bit about that and how the evidence shows its support of the creation or intelligent design models. Of course, we're talking about origins in this channel. How do we get here? That's always been a great debate, probably will still be debated for a lot of years yet. But the idea here is that people want to know how did we get here? Is there a purpose and meaning in life, or is it all just by chance? Now, on this channel, if you have been here before, you'll know that we use this model evidence approach when we talk about origins, because origins is a historic event. You can't actually go back and see evolution occur because it takes millions of years. And of course, creation, you can't go back to the original time of creation. So it's a historical event, but that doesn't mean we can't talk about it scientifically. We use this model evidence approach. The idea here is that origins is the thing that we're investigating. And so what we want to do, there's two ideas. Either we got here by chance, the evolutionary idea, or we were actually designed or created, and that's the creation idea. So these are two models that we have to kind of try to describe how we got here. And we can take a look at scientific data today, evidence that we can find in the laboratory, and we can test and repeat, which that's an important thing in science. Observable and repeatable are really the hallmarks of the scientific method. And so if we can take a look at the data and we see how the data fits in with these two models. And of course, if the data all basically support the evolutionary model, we'd be kind of foolish to believe in the creation model. And of course, the same way, if a lot of the evidence supported the design or the creation idea, then we might not really want to go with the evolutionary model. So it's all based on evidence, and we're trying to find out which model has more evidence. Now, if you've seen some of our other videos already, we've already talked about the evolutionary theory, and we've talked about the theory that says that small changes in, like, say, the genetic makeup of critters, in other words, microevolution, will lead to major changes, which we call macroevolution, over time. And that's the key thing about evolutionary theory. It's not just these little small adaptations or microevolutionary changes, but you have these macroevolutions where you have one animal change into completely different ones. And of course, that happens, according to evolutionists, through natural selection and random mutations. So you get a mutation and you get some small mutations over a long period of time. And then nature selects for the fittest, and then they survive and they get some more small mutations. And over a long period of time, you go from microevolution, or what we would maybe call adaptations, small adaptations, to macroevolution. And this is the key, because some people define evolution as just these smaller changes, but the evolutionary theory has to actually go from changes in major body forms. So you'd have to go from invertebrates to vertebrates. You'd have to go from fish to, like, say, amphibians, or maybe amphibians to reptiles, and you'd have to have maybe reptiles to mammals, or reptiles to birds, mammals, and eventually to man. And so you have these major body changes, these forms, these types. Over here, these are all basically dog types of critters. And so those are smaller changes, and we know those occur. I mean, we can do these, and just think about all the different dogs that we have. We just, by selective breeding, and we can show that in nature some of this can happen. But the question is, did these major forms change from one to the other? In other words, can macroevolution actually take place? So what we want to do is to look at developmental biology today, real briefly. Developmental biology is basically, kind of like embryology. You start out with a cell, and then you eventually get many cells, and then eventually you get these small forms of animals that eventually develop into the adult form. So we want to look at basically three items in developmental biology today. and see how they stack up with this debate between creation and evolution. So the first thing I want to talk about is body plans, those major body plans that we are talk about often in macroevolution are decided early in the development of an organism. And this comes a lot, we initially found a lot of this information from stem cell research. You may have seen diagrams like this where you know that if you fertilize an egg and then it divides into two, and then those two divide into 4 and 8 and 16, and eventually you get like a blastula, and then you get a gastrula, and then a neurula. And if you've done any kind of embryology at all, you probably recognize that. Well, why does a body plan have to be early? Well, the idea here is that when you first have the zygote, that has all the directions, and then it starts to divide. And you have through these cleavages, these different cleavages, things divide into 2 and then 4 and then 8 and so on. Okay. And eventually, like this cell right here, this one in the top left here, eventually that's going to develop into a whole bunch of these cells. And then these cells are all going to come from those cells. So basically, we're just dividing cells. Well, once you get to the blastula and the gastrula stages, once you get to those stages, you already have some of the body plan already developed. You already know where the ectoderm is and the endoderm is and where the blastoceles are. All of this is basically determined by the time you get to this place right here. And so obviously, if you're going to change something, it has to be early on in development. In other words, if you're going to change a body plan, it's going to have to happen up in this area pretty quickly, because by the time you get to here, these cells are so specialized they can't go back. They're not like stem cells anymore. They're basically cells that are already specialized. So if you're going to change a body plan, you got to do it early. The key here is early in development. The second thing that developmental biologists have shown us is that early mutations are lethal. Now, remember, we just said that you got to have early mutations to change a body plan, but evidence is starting to mount that early mutations are actually lethal. Two scientists that did a lot to help us to understand this process is the Michelin Villard and Weisches. They were Nobel Prize winners in science. And basically what they did was they worked with the Drosophila fruit fly. And, as if you know much about Drosophila fruit flies or fruit fly embryology, you get a fertilized egg, and then eventually those parts of the egg start to divide and develop. And eventually you get the pole cells, and you get like ectoderm cells on the outside, and then you get the blastoderms and stuff. And eventually you get these different parts, like the segments in terms of an embryo and that kind of thing. And eventually, this part becomes this part and so on. Well, how do we find out all about that? Well, that's where those two scientists came into play. What they did was they used x-rays, and they would mess around and destroy certain genes. And what they wanted to do was to take certain genes and mess with them and irradiate them, and then kind of mess them up so they could see in the adult fly what they messed up. And so basically, here's a Drosophila chromosome, and it's one of the larger chromosomes. They actually mapped the whole genome of the fruit flies, and they did this by x-rays. And what they did was they x-rayed different things, and they would find different changes. So, they might x-ray one thing and mess up one particular gene, and the eye wouldn't develop, or maybe the wings wouldn't develop, or hairs on the back of their head, or whatever. Now, what was really interesting about what they found, and they got the Nobel Prize for this, they mapped the whole genome of the Drosophila, and so they got the Nobel Prize. Very intense work for several years. But what often isn't talked about is that when they were doing this, they found out that the mutations that are expressed early in the development are inevitably lethal. In other words, if you had mutations that occurred early in the development, of the embryo, they were lethal. Well, remember, we're trying to get early, we're trying to get early mutational changes in order to get changes in body plan. So what that means is that the type of mutations needed to generate a whole new body plan, in other words, during early development, if you do it during early development, you actually kill the animals during their embryological development. In other words, it's lethal. And so, you can't get those early body plans to change because the early mutations actually kill the animal. Now, the third item here we want to mention is the disruption of DGRN, and they're lethal. If you disrupt those are lethal. You say, well, what in the world are DGRNs? Well, the guy that first came up with this, and really probably was the expert, spent more years, and I think he just died a couple years ago, but he spent more time than almost anybody studying this, was Eric Davidson. And he realized that this junk DNA, there's DNA that for a long time evolutionists have said they didn't have any function at all. They were just left over from previous things that supposedly we inherited from our common ancestors. But actually, junk DNA was actually important in regulating embryonic development. Davidson did most of his work with purple sea urchin, and he mapped the whole genome of that. And so one of the things that he discovered were these developmental gene regulatory networks. That's what DGRNs are, okay? And there were these whole networks of things on the junk DNA. And what they basically did is they produced proteins that act as a signal molecules, and they help control the development. So they turn certain genes on and certain genes on at a certain time, and then those genes make signal molecules that they call for other things to turn on and turn off. So these are like really, really complex things. They're so complex that a lot of people discuss them like they do integrated circuits. Here's just a map of one of the ones that they have actually mapped, and you can see there's different types of inputs and there's different types of outputs, and they go depending on if they get signaled for or they don't get signaled for, which is really much like a integrated circuit. Now, what Davidson found was that these are stubbornly resistant to change. I mean, they don't change because they're so complex, but you can change them. And if you do change them, what happened? Well, there was a catastrophic loss of the body part or a loss of the viability altogether. He went on to say that there's always an observable consequence if a DGRN subcircuit is interrupted. So if you interrupt that somehow by some type of change, these consequences are always catastrophically bad, which means that they're not very flexible at all. So not only, what are we saying here? So not only, the body plans have to develop early in development, but if you have those occur early in development, they're always lethal. And not only that, but if you have mutations occur in these regulatory networks, besides these other things, if those mutations occur there, they are also always lethal. And in order to get a mutation to work, you've got to have these networks. So what are we saying? Well, basically, the experimental evidence is indicating that random mutation and natural selection can't produce new body plans with new information because it has to occur early, but when it occurs early, it's always lethal. And so even though you might have these microevolution or adaptation types of things, and we know that those happen in the laboratory, but we have never seen this thing going from one body plan to another body plan. We see it in the fossil record, but we don't see these changes to make these transitional forms between these in slow, gradual jumps. And the reason probably why we don't is because of this information in developmental biology, that things have to occur early, but if these changes occur early, the body plans basically are scrapped and the embryo dies. So what are we saying? Well, that evidence really supports the creation or intelligent design model. In other words, the evidence, if you look at the evidence, it doesn't really support this slow, gradual change from one small thing to another, and eventually those turn into big changes. Quite the opposite. If you are forming new body plans and new parts, the, trying to get that to occur by changing uh, the genetic information is basically almost, it's basically impossible. It's lethal. So, a really important concept, early mutations have to be there for changing the body plans. But when you have early mutations, it not only destroys the genes themselves, but it also destroys those junk DNA genes that also control those regulatory networks.